Yeah. So as I said earlier, I will have Dr. Biden go over the quiz next class, just because of the logistical things we had to go on with the quiz. But today we're going to talk about protein trafficking and immunity. So previously we've already learned about DNA replication, transcription, DNA RNA, and also translation of RNA into polypeptides. So today we're going to be talking about how these proteins are trafficked or moved to their final destinations. The locations of final destinations are very specific to the protein's function. Also, it's unrelated to the function. I'll do that. But they can go to the membrane of the cell, the extracellular environment, or other organelles. And so we're going to be talking about how these proteins are trafficked and also the context of Noonan syndrome. So today I'll be talking about Noonan syndrome, introducing it, but we'll only get through part of the lecture, so we'll finish it next class. So Noonan syndrome is a congenital disorder. Congenital means that you're born with it. And some symptoms of Noonan syndrome include microcephaly, which is a large type. We talked about Down syndrome, and which can be brachycephaly, and then trisomy 18, which causes microcephaly. So brachycephaly is just a smaller type. The microcephaly is like a very small head. Another symptom is hypertelorism. That consists of longer limbs. You can kind of see that in this image with the child. They have really long arms. And then it can also result in a shorter legs and neck. So it actually has, the incidence is 1 per 1,000 to 2,500 live births, which is the same incidence as cystic fibrosis. So it's not as rare of a disease as you would think. It just doesn't get a lot of recognition on this. There are some associated complications with Noonan syndrome, including cardiac complications, but when they're addressed with the correct surgery or medication, people with Noonan syndrome can live to old age. So the etiology of Noonan syndrome is a defect in the RAS-MAPK signaling cascade, which we will go over next. This is an example of someone who has Noonan syndrome. You can see the short web. Okay, so now we'll talk about the RAS-MAPK signaling cascade. So overall, a cascade, whenever you hear that in terms of like biology, that basically means one event causes the next one. So it's like a downstream event. And so it begins with activated RAS, which is up at the top. Yes. And so this is a monomeric G protein, which we'll talk more about on the next slide. But basically, RAS activates the protein kinase RAF. So RAF is another enzyme. And so a protein kinase is an enzyme that phosphorylates or adds phosphate to the target protein. So basically, these enzymes are phosphorylating another enzyme that allows it to become activated. And so as we talked about before, there are 3 amino acids that can be phosphorylated. There's serine, threonine, and tyrosine. In this case, RAF is a serine-threonine kinase. And so basically we'll only add phosphate to either serine or threonine amino acid. And so when RAF is activated, then it will activate another kinase, which is MEK. So lots of kinases is basically just a whole stream of enzymes that will, once activated, activate another enzyme. And so MEK is a dual specificity kinase. So it can phosphorylate both tyrosine and serine and threonine amino acids. And so that's why we have this like branch in the middle, the dual specificity, because it can activate one set of amino acids, which is the serine and threonine, or the other set, which are tyrosine. And so once MET is activated, then that will activate MAPK, which is MAP kinase. And so these are class of proteins that aren't just one single protein. They have different names. There's KRAS, HRAS. It's the same for all the enzymes in this cascade. But it's not just like one name of an enzyme, it's like the category. Does that make sense? Was that confusing? Okay. And so MAPK, it activates a transcription factor. So that's the last row that we have here, the activated TF. TF is transcription factor. And so this activated transcription factor will target downstream DNA and promote transcription. So in summary, we go from GRAS, once activated, to GRAF, and then MET, MAPK, and then activated transcription factors. And so the phosphorylation of these enzymes stabilizes the transcription factor. It also increases the binding to DNA. It promotes mobilization into the nucleus for promoting transcription. So that's what phosphorylation of the transcription factor does. Does anyone have any questions about this? Yeah.
Usually the dual specificity.
Yeah, so I wasn't very clear with that earlier. Basically with kinases, there are two different categories. Because we have 3 different amino acids that can be phosphorylated, and they're split into 2 categories though. So there's the serine and threonine, that's one category, and the other category is tyrosine. So that's what the dual specificity is that it can activate both of those categories. But typically kinases are either serine and threonine or they're tyrosine.
Usually split is
It's so, for example, like this side, the left side would be serine threonine, and then the other side would be tyrosine. That's just because there are a bunch of different MAPKs. It's not one. It's like the same. Questions?
Yeah, this little split is confusing me too. So the first split is because one of these activated or kinases is going to do serine threonine, and the other one's going to do tyrosine. But then, like, why do you split again at the next and the next as well?
Don't overthink the splitting. I think this is just his way of showing that it's like a downstream effect. I think you do need to know that MET is dual specific, right? So it has those 2 categories. But then in terms of like the splitting downstream, like this yellow box and the green box, it's saying that there's multiple MAPKs that can be activated, also multiple transcription factors that can be activated, depending on which one is activated. Does that make sense? Okay, so we talked a lot about GRAS. So it's a monomeric G protein. So previously we learned about trimeric protein linked to GPCR, but GRAS is a monomeric, which means it has 1 subunit. So remember, heterotrymic is there's 3 subunits to it, and hetero means they're all different. So that's why you have that alpha, gamma, and beta. But then GRAS is monomeric, so it only has the alpha subunit. And then the alpha subunit binds to GTP, and then that's how it's activated, and then it's deactivated when GTP is hybridized to GDP. And so heterotrymic and monomeric G proteins are constitutively localized to plasma membrane. So that means that there's a modification to the proteins that restricts them to stay on the plasma membrane. They won't be able to go into any organ else. Okay, so now that we have a little bit of background about Noonan syndrome, we're going to get into trafficking of proteins. So basically, we know that transcription occurs in the nucleus, and the mature mRNA is then shuttled to the cytosol, and that's where translation occurs. And we'll be talking more in depth about the organelles that are responsible for the trafficking of the proteins. So first, this slide is about endocytosis and membrane cycling. So this is like a big overview of the endocytosis and exocytosis that occurs in the cell. So endo, right, that means into, exo means out of. So you have your nucleus on the left side, and then you go to the rock-ER. Rock-ER is studied with ribosomes, where the ribosomes are responsible for the translation of mRNA into a protein. It's directly adjacent to nucleus, which is important just because closer to the nucleus, right, the faster the translation occur. And so there are two styles of protein transport. Exocytosis, that's leaving the cell. So that's on the top portion of this image. It's also called anterograde transport. So it goes from the rock-ER to vesicles and then out. And vesicles, basically, have you guys been over what vesicles are? A little bit? Okay, cool. So vesicles will just help transport between organelles, and then once at the Golgi apparatus, vesicles will form to release to the extracellular space. And to do that, they fuse with the membrane, and then it's released to the extracellular environment. On the other hand, we have endocytosis, so that's things, those are things that stay in the cell or come into the cell. So we talked about botulism toxin, that is endocytosed. And basically it's the opposite of exocytosis. So it's called retrograde transport. You have an evacuation of the membrane, and then the endosome is formed, and then it will bind with a lysosome. And then the lysosome will either degrade what is brought in, or it will release it into the cell. So basically you're going from the outside the cell into the rock. Yeah.
What is the difference between the lysosome and the late endosome?
I think that's different stages of how an endosome is the lysosome. We'll talk about it a little bit more, how lysosomes are formed. I don't know off the top of my head what distinguishes exactly between the late endosome. All right, so we talked about vesicles, so now we're going to talk about how they're formed. So we have a clathrin-coated pit. And so the invagination of the membrane that I talked about, that's mediated by this clathrin-coated pit. And this is a scanning electron micrograph of one of the pits, on a scale of 0.1 micron. And so this is what we would see if we removed the membrane. So basically, if you had, like, here, I can't zoom in, but you have a membrane that's encasing the endosome, and so if you removed it, this is what you would see. And so it goes and creates a cage, which pinches off, and it was being endocytosed. And so I'll go into more of that on this slide. And so clathrin, it's pretty unique, it has a pretty unique structure. It's a heterohexochromic protein, and it forms a triskeleton. So basically that just means it has 3 arms that curve. You can see that in both of these images, like this pink structure. And you have 3 heavy chains and 3 light chains. So the heterohexochromic structure allows it to assemble into a cage-like structure. As you can see on the right side, there's that sphere. That's what we call a cage. And so that forms when the clathrin links together. And so this will coat the entire surface area of the vesicle and mediates the formation of the endosome. This is initiated by ligand receptor binding events. So there are adapter proteins. And so adapter protein basically is protein that physically interacts with two things and it brings them close together. It doesn't interact chemically with things that it's like bringing together. It just brings them in close proximity. And so the adapter protein interacts with the receptor and the clathrin. And so it allows for the assembly of the clathrin coated vesicle near the plasma membrane, which is essential for creating an endosome. And so this is an example of receptor mediated endocytosis, because you depend on the binding of that receptor to endocytose or bring something into the cell. And so this is like a third of the adapter protein that I talked about, and how basically it just brings the clathrin and the receptor together. Okay. So endosomes, these are vesicles that are inside the cell. So once the membrane is pinched off and you have that, I guess you have the vesicle inside the cell, it's called an endosome. And so to address your question, once the clathrin is released, the endosome can mature, and that's how it becomes a lysosome. So to form an endosome, the first thing that you need is the ligand receptor bonding. So the receptor helps bring proteins on the plasma membrane. The adapter protein brings in clathrin and begins creating the cage that we talked about to form an endosome, but it's not ready yet to pull off and form that vesicle. So to do that, you need another enzyme called dynamin, which is in this blue box in the middle of the image. So this is GTPase, which means that it hydrolyzes GTP to form GDP, and by hydrolyzing GTP, it forms a bridge across the coated pig. So basically, to think about it, the diamond is essential because when you like pick an apple from a tree, you kind of twist it to get it off the tree. You can't just kind of yank on it. So that's kind of what diamond is. It's twisting. Pinching off the membrane so that you can form the vesicle. And so now you have clathrin coated vesicle inside of the cell, and the clathrin will eventually be released and recycled. And then there's a proton pump that helps to decrease the pH to disrupt the ligand binder interaction, and that's how your ligand is released. And so sometimes your ligand is released on the opposite side. So you could go from like the basal side of the cell to the apical side, and this is called transcytosis. Anyone have any questions so far? Yeah.
So the coding is
Right. Is that your question?
Yeah.
Okay. Yeah, the coding is clathrin. Okay. Yeah.
So when the vesicle like begins with the endosome, that when the coding is that what you said?
Can you say it one more time?
What point is it at the ligand part of the?
Yeah, so the ligand, right, that's inside of your vesicle, and that's what we want to either be released into the cell, or degrade it. So that occurs once the vesicle is uncoated. So once the clathrin disassembles, then you're able to either convert the endosome to a lysosome, or to do transcytosis and release the ligands elsewhere. Any other questions? All right, so now we'll talk about protein synthesis, export and import. So we looked at endocytosis, and so we'll get exocytosis of newly synthesized proteins. So there are different ways to make proteins for organelles, membranes, or the extracellular space. So they're called importing, with types of imports. So first is co-translational import. So this means that during translation, as the new peptide is being formed, it's also imported into the ER. So this happens at the same time. So as you're building the peptide, it's also being put into the endoplasm. And this is specific for proteins that employ extracellular space, cell membranes, and liposomes. And then you have post-translational import. This means that once the peptides are fully translated by free ribosomes, they're transported to their final destination. So these will go to the nucleus, the mitochondria. And it's determined by signals embedded in the amino acid sequence of the newly synthesized protein. So I'll go into more depth about these on the next few slides. So protein trafficking, in the absence of any signal or targeting sequence, proteins are made and they remain in cytoplasm. And this is typical for most of the proteins in cells. The RAS-MAPK cascade, that's an example of proteins that don't have signals and they'll be trapped anywhere. They'll just stay inside the cell. Co-translational import has a signal sequence. So the presence of this sequence encodes either the default destination for exocytosis or to the plasma membrane. And then if you have targeting sequences, they can direct them elsewhere. But basically, when you have a signal sequence with co-translational import, they'll either be exported, the protein will either be exported from the cell or goes to the cell. And so you have to have an interference with the targeting sequence that will put it or direct it to a different location. For post-translational import, you have two different sequences, the nuclear localization sequence or NLS, and the mitochondrial targeting sequence or MTS. They're pretty straightforward based on their name. The NLS target proteins for import into the nucleus after translation, and then the MTS target proteins for import into the mitochondria.
So both types of import have their own signals.
So they have their own sequences, but they're not all called signal sequences. It's only a signal sequence with co-translational, or post-translational, either the NLS or MTS. Okay. Oh, and then proteins that are smaller than about 40 kilodalton will freely diffuse between nucleus and cytoplasm. This is an information for later. I don't cover it, but we will on Tuesday. Okay, so now we'll go through the co-translational imports. I guess just entire process. So co-translation import. This is the one where the translation occurs at the same time as doubling the protein to the rough ER. So first we have SRP. That is it's a protein. So the protein binds to the ER signal. ER is the signal. And so it binds to that signal sequence on the polypeptide chain. So that's what's happening in the first picture. In the second picture, the SRP also binds to the SRP receptor. So this receptor is found on the membrane of the rubber. And so the ribosome also binds to the RUF-ER receptor, I believe. And so this stabilizes the entire structure of the ribosome with peptide growing out of it. So basically, you should stabilize it and keep everything localized to the RUF-ER. Next, third step, GTP binds to the SRP receptor. And when the SRP binds to the signal sequence on the polypeptide chains, translation is momentarily paused. And so when GTP binds to the SRP, it's hydrolyzed to GDP, and then the peptide is put into the core protein. So the core is this purple thing. So this core allows the protein to enter the RUF. And the next step, once the GDP is hydrolyzed, the SRP is released. And then translation, step 5 just continues. The peptide is elongated and it is translocated into the RUF-ER lumen, or the space between the membrane. Then last, there's a signal peptidase that cleaves the fully translated peptide and releases it into the lumen of the RUF-ER. Any questions about co-translational import? I think just make sure that you know the steps and when something binds, what is the next step, essentially. I feel like that's something that you guys maybe put it on. All right, and so this is just an image of what the real RUF-ER looks like. And so this is a ribosome membrane. And this is super zoomed in, and so this is what a core protein looks like. So there are 3 subunits, and it's fairly dynamic, which means that it can move around, and this is important for when transmembrane proteins are produced, which I talk about on the next slide. Okay, so for transmembrane proteins, this is called stop transfer or topogenic sequences. So in the beginning of the semester, we learned about the amino acids and how a sequence of 20 hydrophobic amino acids indicates that it's a transmembrane protein, right? Because when you have a transmembrane protein, it has to have a portion that will interact favorably with the membrane. So for the co-translation of a transmembrane protein, there is a stop transfer or a topogenic sequence. So again, those are the 20 hydrophobic amino acids in a row. And so once they're recognized, which is kind of panel 2, the dynamic core protein releases the peptides. That's why it's important that the core is dynamic, because it's able to release the peptide that's in it. And so basically the hydrophobic amino acids do not want to be near the lumen of the RUF-ER because the lumen is hydrophilic, right? We have hydrophobic amino acids. And so it will shift to the membrane of the RUF-ER. And so the ribosome will no longer be sitting on top of the membrane. Now it's in the cytoplasm of the cell, and then you create the rest of your polypeptide. So basically, once it recognizes the 20 amino acids, then the rest of the protein is just formed outside of the RUF-ER. and then it's released from the core protein. And again, this is just to accommodate that hydrophobic does not mix with hydrophobic. Okay, so now we'll get into post-translational modification in the rough ER. So once translation is complete, newly synthesized transmembrane or luminal polypeptides will undergo 5 principal modifications in the ER. The first one is the formation of disulfide bonds. This is done by the protein disulfide acetylase. And so if you guys remember, forming disulfide bonds will occur in oxidizing environments, and the rough ER is an oxidizing. Other modifications that will undergo include proper folding, specific proteolytic cleavages. Proteolytic means it breaks up the protein into smaller chunks. Assembly into multimeric proteins, and then addition and processing of carbohydrates or so now we'll talk about what glycosylation is. So, glycosylation can come in a variety of forms. The two that we'll focus on are N or O-linked oligosaccharides. So, polysaccharide means that there are many links, and then oligo, we would say is like a few to a dozen. That's like the numbers. So for O-linked, this is on the left side of the figure. Basically, the sugars are linked to the oxygen of the hydroxyl group, so the hydroxyl group is the OH group. And so basically you have like examples of different O-links. You don't have to memorize any of those, you just need to understand that for simplicity's sake, O-link means that it binds to the oxygen of a hydroxyl group. So that would be styrene. On the other hand, also O-linked oligosaccharides can be very short. And on the other hand, you have N-linked. So by the name, also on the right side of this figure, by the name you might really infer that N-linked means it's linked to nitrogen, and they can be more complex. So this is a binding to nitrogen of asparagine. And so different branching events can occur based on how the bonds form. And so the development of the bonding determines if it's an alpha or beta link. This is kind of like a biochemistry concept. It's not really important that you know like alpha versus beta. It's just important to know that they can N-linked oligosaccharides tend to be long and highly branched. So basically, think of it like 3 you'll have branches. O-linked, I would say, is like a single stick. Does anyone have any questions? Yeah.
Can you go back to the last slide?
Good.
Yeah.
And so this bottom photo is an example that a O-link. Yeah.
So are these the different types of glycosylations that happen in the first translation modification?
Yes. Speaking of glycosylation, this is kind of how it happens. So basically it will start in the ER, the lumen of the ER, and then it's further elaborated in the Golgi. So basically glycosylation will start in the rough ER, and then you'll go to the Golgi, and it will continue to add more sugars. And then basically in the Golgi, there are lots of different compartments where there are different enzymes, and so sugars will be added in a stepwise manner. And there are a lot of sites on proteins for where sugar modifications can occur. So this can result in incredibly complex proteins. So you get glycoproteins or proteoglycans. And so something to highlight is glycocalyx. So the glycocalyx, these are all the sugar modifications that occur on the plasma membrane. And so just to paint a picture of how glycosylation can affect proteins, you have two identical copies of protein A, and when they go through the glycosylation process, it's so complex that by the end of the entire glycosylation process, like once it gets out of the Golgi, they'll likely be very different in structure. They may have the same function, but they'll just be very different. It's based on where the sugars are added. Okay, so this is an application of glycosylation. And so we talked about Western blot before. This is an example of Western blot. And so when you have a bunch of sugars on proteins, it's very significant both in research and clinical applications. So for this Western blot, I believe it was a student, maybe a PhD student, who was using mono and polyclonal antibodies to detect GDF11. I don't know what GDF11 does. So basically you're just trying to detect this protein. And usually with Western, you're trying to get a super clean band. But they ended up getting like a lot of bands and it's really messy. And if you were done Western, this would be hard to interpret. So they're trying to figure out, you know, so they have a dirty antibody, they just really bad at doing Western blots. And so it actually happens with two different antibodies, the messy Westerns. So on the left is one antibody, on the right is another. And so the researcher ended up using a D-glycosylating enzyme. And by adding that to their Western, they were actually able to get a clear band. And so basically that just showed that there was so much sugar on the protein that it blurred the band of the Western. And also it was really hard to see the signal using a monoclonal antibody until it was de-glycosylated. So moral of the story is glycosylation can hide and block epitopes of interest. And then another application of glycosylation is with blood. So alleles are different versions of the gene, and so there are 3 alleles for blood. There's A, B, and O. And so the allele for gene of interest that we're looking for codes for an enzyme that will glycosylate blood. So for people who have the A allele, which is A blood, they'll have GalNAc transferase. Basically, they will have this N-acetylgalactosamine added to their base protein. For people who have the B allele, they have galactose transferase, and that will add a galactose to their base molecule. And then for O, they actually don't have an enzyme, so they don't have any sugar. And so if you have 2 copies of the O allele, that means that you can donate blood to anyone. And so the positive or negative of like your blood, if you're like A positive, B positive, all that, that's related to protein or antigen, it's the RH, that basically is not related to sugar. So your type of blood, so whether it's A, B, or O, that is related to which enzyme you have that will glycosylate your protein. Does that make sense? Okay, and then the last thing I will go over will be the sugar-based protein targeting lysosomal enzymes. Basically, this is how you get luminal proteins in Golgi to localize to a lysosome. So you want to localize acid hydrolases that will chew up stuff into the lysosome. And you want to contain those in the lysosomes because you don't want degrading things at random in your cells. And so there's a post-translational post-translational modification that occurs on the lysosomal hydrolases. That attaches the mannose-6 phosphate. So the enzyme that conjugates sugar to the enzyme, recognize the signal cache on the hydrolase and adds the mannose-6 phosphate. So basically there's an enzyme that will recognize the sequence on the lysosomal hydrolase, and that will cause the addition of the sugar for the mannose-6 phosphate. And so once this sugar is attached to the hydrolase, then it's recognized by the M6P receptor, which is facing inward towards the lumen of the Golgi. And so this assembles on the membrane and it creates a codomer. It's not coppering, but it's basically the same idea that you have a codomer that coats the hydrolases, and then they flood it off, and the codomer is recognized by other trafficking proteins, and they use vesicles with lysosomes. And that is all that I had prepared today. I know